So now that we have a sense of how hearts function, kind of broadly speaking, it's now time to start diving in and exploring different circulatory structure alternatives. So we've mostly focused on mammalian and bird circulatory systems so far. So now we'll look in greater detail at the circulatory structures of fish, the amphibians and non-bird reptiles, and invertebrates. And I'm going to break this topic up into a series of shorter videos so that you can kind of focus on each circulatory system by itself, and then potentially break this up in any fashion that makes sense to you. Before we dive into looking at specific circulatory alternatives, I just want to briefly revisit some of the equations that we've examined for understanding the relationships between pressure, resistance, and the overall flow of blood through a circulatory system. This is going to become important towards the end of our exploration of circulatory structures, but just want to get these equations back in your mind now. So we've looked at the Hagen-Poiseuille equation as an overall characterization for blood flow rates, right? So that's related to pressure difference, the size of or diameter of the blood vessel, how long that vessel is, and then there's a couple of other parameters in the equation. We can actually also characterize a phenomenon known as vascular resistance. And vascular resistance, in a lot of ways, is sort of the inverse of the Hagen-Poiseuille equation, right? So it's related to the length of vessels divided by the vessel diameter, okay? And then, we could rewrite the Pn minus Pout as our delta P, or pressure gradient. And that gives us an alternate version of the flow relation, which is basically a function of the pressure gradient divided by vascular resistance. So we'll see this within a couple short videos. Okay, so looking at alternate organizations across different animals, your textbook has this comparative figure to illustrate different circuitry in different types of organisms. I've spent a lot of time scrutinizing these circuitry diagrams really closely, and there's certain aspects to them that can be a little bit confusing, and it may take some time to study these circuitry diagrams to really make sense of them. So along with showing you the textbook versions of the circuitry diagrams, I'm also going to show you an alternate version that I re-illustrated some time ago. And in either case, I'd recommend either drawing your own diagrams or spending a decent amount of time reviewing how all of these are structured. So we'll start out now exploring organizations in fish, and then in our next videos we'll look at these other taxonomic groups. So fish have a very simple circulatory circuit that has just a single pump. And to get this to make sense, let's look back at the circuitry diagram for the mammals and birds. That's on the left here. Right, so in ourselves and in birds, if we start on the left side of the heart, right, with the left atrium, left ventricle, right, that portion of our heart pumps blood through our systemic circuit. Then blood returns to the right side of the heart, and it gets pumped through the pulmonary circuit, through the lungs, where it gets reoxygenated. Then it comes back to the left side of the heart, and that oxygenated blood then gets delivered to the systemic circuit. So let's look at the fish diagram here now. So here's the fish heart. We've got deoxygenated blood from the heart that's going to get pumped through the gills. At the gills, that blood's going to be oxygenated, so now it's fully oxygenated. That's going to be delivered to systemic tissues, and then from systemic tissues, that's going to return back to the heart. So in fish, there's just a single pass through the heart, and between heart passages, that blood passes through gills and it passes through systemic tissues. So look at these, think about them for a moment. And again, remembering that speed is a little different from flow rate. So we're just thinking about relative speeds or velocities of blood moving through tissues here. What I want to ask you is, what do you think the implications are going to be for the relative speed of blood for birds and mammals compared to fish, and why? Take a second, pause the video, jot down a note, what are the implications going to be for fish compared to birds and mammals for relative speed of flow through the circulatory system. So now, having taken a moment to think about those implications, let's think about what we know about the total cross-sectional area of these different aspects of the circulatory system. So at the heart, we have a relatively small cross-sectional area. Blood flows quickly through the heart. When we reach the gills, in both cases, at our gas exchange membranes, blood vessels need to branch out to the level of capillaries. So very small individual vessel areas to maximize gas transport, maximize release of carbon dioxide and diffusion of oxygen into the capillaries, right? So everything needs to branch out, and then past the gas exchange membrane, blood vessels are going to start joining back together into larger vessels again. Right, so if we have a lot of branching out at the gills, total cross-sectional area is going to go up. That's going to translate into a loss of speed and a loss of pressure across the system. So in fish, blood's going to go through the gills, lose speed, lose pressure, then go through systemic tissues where there's going to be a second branching and then rejoining event before coming back to the heart. So I think hopefully this shows you that the overall speed of blood through a fish's circulatory system is going to be a lot slower than the speed of blood moving through mammalian or bird circulatory system, because there are two different opportunities to increase pressure in the system, in the mammalian system compared to in the fish system. So now here's a diagram of the physical arrangement of circulatory structures in fish. And you don't need to know this in excruciating detail for my purposes, but I think it's just useful for kind of understanding the events that are taking place. So we'll start out at the fish heart. Here is our fish's heart. And the heart is going to pump blood to the ventral aorta. So ventral is anything that's on the lower body surface. And at the ventral aorta, this blood is all deoxygenated. So in this drawing, the blood's black. At that point, blood's going to perfuse or flow through the gills, right? So we're going to remember countercurrent exchange here. And at that point, it's going to become oxygenated. It's going to recollect in branchial vessels. So that's a set of blood vessels that are along the dorsal surface of the fish. And that dorsal aorta is then going to carry blood to the tissues, right? So it's illustrated here, blood to the brain, blood to central organs, blood to posterior organs. At the tissues, we're going to see gas exchange. That blood is then going to be deoxygenated, and the great veins here are going to return that blood back to the heart to pass through the system again. All right, so let's talk about some problems for fish. So as I emphasized earlier, the heart has to generate enough pressure to get blood through the gills and also through the systemic vasculature. So the major pressure source in the fish heart is a single ventricle. So that's illustrated down here. And so just to kind of walk you through the pathway of blood through the fish heart, we're going to come in through the sinus venosus. Fish will have an atrium, then blood will pass into a ventricle, and then it will pass out through the conus arteriosus, or in some fish, the bulbus arteriosus. And that distinction I'm not super concerned about for our purposes, but essentially in some fish, the conus arteriosus facilitates additional pumping. And in other fish species, the bulbus arteriosus is closer to what we might think about for our aorta, okay? So in addition to all of this, so our ventricle is a major source of pressure. Fish are where we observe spongy myocardium. So the oxygen source to supply the fish ventricle comes directly from blood that's passing through the heart lumen, okay? So some blood has to remain in the ventricle to go through oxygen exchange with heart tissue, okay? So this might seem like not an optimal arrangement to have. However, in a lot of ways, this doesn't really have major negative repercussions for fish for a range of different reasons. First off, if we think back to the relative oxygen demands of fish, fish don't actually need as much oxygen as terrestrial animals do. They have lower metabolic rates. We'll see a lot more about this fairly soon. So they don't really need as much oxygen. So that means that they can get away with smaller hearts, so much lower cardiac output, and generally lower arterial pressures. So the maximal pressure you could ever observe in a fish artery is going to be about 12 kilopascals. And that contrasts to, if we think about human arterial pressures, 16 kilopascals is roughly equivalent to 120 millimeters of mercury. That's pretty high for humans. But again, this is the maximum that a fish heart could ever sustain. And all this said and done, heart characteristics and fish physiological performance are correlated with the fish's degree of physical activity. So, these things, they're not set in stone. They can also be phenotypically plastic. So in other words, cardiac characteristics can change depending on the level of activity of the fish. And so just to kind of give you some examples of this, fish like salmon that we saw at the beginning of the semester and tuna do have some compact myocardium that gets oxygenated blood around the exterior of the heart. And tunas, again, kind of are exemplar for a number of reasons. So they pump their blood much more rapidly, and they have twice as high of a weight-specific cardiac output at rest and very high blood hemoglobin concentration. So that all suggests tuna have higher oxygen requirements, and they've adopted a couple of different strategies on an evolutionary timescale to facilitate better oxygenation, tissue oxygenation, relative to your generic trout. Okay, and I've also put this picture here from your textbook on this slide for salmon heart characteristics. So at the beginning of the semester, the video that we watched illustrated salmon migrating upstream to spawn. And not all salmon migrate upstreams that are equivalent in the difficulty of the challenges that they pose, right? So you can imagine, some rivers, like if you go up to the Hudson River, we don't really have major waterfalls. That's a fairly easy migratory pathway for a fish. In contrast, if you think about other bodies of water that do have more extreme waterfalls, those are going to be physiologically much more challenging for fish to traverse. So our x-axis in this graph is an index of a fish's migration effort that's needed to reach the spawning area for that body of water. And this is for salmon. And then the y-axis is the percent of the salmon's heart that consists of compact myocardium. So what I hope you can see here is that fish that have a relatively easy time migrating, so on the Weaver or Harrison rivers on the left-hand side, have a lower percentage of compact myocardium relative to fish that have more hardcore migratory efforts, such as on the early Stewart River. Okay, so this is an example of local adaptation to the specific spawning conditions across these rivers. And so clearly there's some room for both phenotypic plasticity changes over the lifespan, but also local adaptation to those conditions impacting heart function. Okay, so let's take a look at this one more time, just using a little cartoon I created. Right, so in general, for thinking about fish circulatory function, blood's going to arrive at the heart. It's actually deoxygenated. It's already passed through the systemic circuit, through the atrium into the ventricle, which has spongy myocardium with varying amounts of compact myocardium depending on the species and the conditions. Then it's going to pass through the conus or bulbus arteriosus, through the gills, and there's going to be major pressure loss at the gills. And then oxygenated blood will then travel through the systemic portion of the circulatory system. All right, we'll pause there in this video, and in the next video, we're going to talk about some really interesting alternatives to this circuitry in the air